Good morning, everyone. Good morning, Antonio.
Good morning, good afternoon here.
Yes, I'm so sorry. I'm in Brazil. So good afternoon, everyone. Welcome to the section 4. So in this section, we'll have some thoughts regarding towards sustainable management strategy. And before we start, I would like to thank the organizers to invite me and also Antonio Vicente to be chair of this very important section. And so to start, I would like to invite Dr. Donato Boccia from Institute for Sustainable Plant Protection, National Research Council of Italy, to speak about the searching for olives displaying resistant traits to Xylella fastidiosa subspecies pauca ST53, experimental evidence and challenges. Thank you very much, Donato, and you can start your talk.
Thank you, Alessandra. Good morning to everybody, and thanks for giving me this opportunity to share some of our data with you in this conference. So as you can read from the title, my talk will be searching for olive displaying resistant traits to Xylella fastidiosa subspecies pauca ST53, experimental evidence and challenges. So these activities have been originated by the preliminary observation done soon after the identification of the outbreak in South Italy, in Apulia. So the observation of evidence of differences in the olive germplasm, because as you can see from this picture, you can see the great phenotypic difference between Oliarola, the autochthonous cultivar very susceptible to Xylella fastidiosa, and Lecino, one that was almost soon identified with the characters of resistance. There are two research lines that have been established since a few years with having the same objective, the same aim. One is the screening for olive cultivar for Xylella fastidiosa resistance. So the first research line has been is going through the evaluation. So I will of circa 100 genotypes that have been planted in experimental plots, and in these plots, we planted Xylella-free trees in areas with high pressure of inoculum. And in these plots, what has been done is going on is the recording incidence and progression of the infection during the time, and recording also the scoring of making the scoring of the symptoms. The other parallel research line is going on in greenhouse, and so far, and what I will present briefly now, we concluded the evaluation of 76 genotype potted. They are potted plants have been subjected to needle inoculation with the bacterium, and also for this we are recorded the evaluation of systemic infections and symptoms scoring. For this trial are exactly 76 selections have been tested, majority coming from the World Olive Germplasm Bank and Breeding Program of IFAPA in Cordoba, so from Cordoba, Spain. Those have been inoculated between March and August 17, and 8 of them were excluded due to plant mortality that occurred at the beginning, but not due to Xylella fastidiosa. They are mostly self-rooted plants of 40-60 centimeters in height, and or the minority grafted 2 years old plants, and are the cultivars of Slovenian origin, provided by Tanja Dreo. Few selections were re-inoculated in 2018 as no colonization was detected. And the strain used for the inoculation is the Dedonno strain of the genotype ST53, growing in BCY PD3 media. And diagnostic tests that have been done, the first is the first check is 1 month post inoculation, when plant are tested at the inoculation point to assess inoculation efficiency, yielding, and that we found yielding over 70% of positive samples. So quite, in fact, Maria Saponari, that is my colleague managing these trials, I think is quite satisfied for this results. Then plants were tested at 10 centimeters above the inoculation point, at over 15 centimeters, and also taking leaves from the apical portions. Symptoms start to appear the earlier at 10-12 months post-inoculation, but only on a very limited number of plants, because the majority started to show symptoms later on, as you will see later soon. We categorized the selection as high susceptible if after 3 years, the infected plants were completely desiccated. Susceptible if 50% or more of the plant canopy was desiccated, and potentially interesting if when we have infected plants with no symptoms, or if the shot dieback appeared limitedly to the inoculation shot. Results on the selection. As you can see from this small table, the rate of systemic infection has been between 16 and 30% in case of 13 selection, between 40 and 55% for 18 selection, and the remaining between 60 and 100% of systemic infection. For the few replicates infected, for the 13 selection, few replicates were infected as S. So susceptible 3 selection, highly susceptible 5 selection, while as potential interesting, we got Lecino that has been used also as resistant control, because we found since the beginning Lecino with the characters of resistance, Ascolana tenera, but the greenhouse, as you will see later on when I will talk about the field trials, the greenhouse results are not completely overlapping with the field results. Docker, and we have in this case only 2 infected plants and no data from the field yet. And Lecce del Corno, another cultivars that apparently potentially interesting from the field data, also again we run a survey in a commercial orchard, in a commercial olive grove, but in that case, this potential interest was significantly reduced because the cultivar show a quite high rate of infection, also developmental symptoms. So you can see here the Lecino shoot dieback only on some inoculated shoots, but mostly the Lecino remain in good condition. In case of Ascolana tenera, only one repetition got infected, showing mild shock dieback, but as I said before, in the field the situation appeared differently. And then you can see some example of highly susceptible cultivar, like you can read here what kind of selection are. And then for the others, we have more or less similar situations, like I will show you quickly some these susceptible selections, subspecies Europea varieta silvestris, subspecies 1 chica, and you can see here into the majority of the trees there were none was potentially interesting. The majority were highly susceptible. Very few, very with intermediate susceptibility. You can see here some example. So in this case, Moraiolo, Moraiolo it is interesting because looking apparently promising for a couple of the first 2 years, collapsed completely in the third year. And similar situation we got also in the field, where at the beginning we considered Moraiolo as a promising cultivar. So you can see some selection of Frantoio also. Well, other selection of Frantoio, Manzilla de Sevilla, Chiquita, and very much quicker and very more clear results we got from Slovenian germplasm, because in this case the difference was probably due to the fact that the plants were older, already two years old when were injected, and were grafted, not self-rooted. So the assay of the greenhouse is challenging, especially when a few replicas become systematically infected. This determines that the phenotypic judgment must be done on a few plants, which is always risky, especially in the case of the absence of symptoms. Our evidence suggests use of bigger plants and use of grafted plants. The reason for grafting, as reported in the poster of Giuliano Lo Console and co-worker, is not clear, and the role of roots is something to be evaluated yet. In the case of the field experiment, I already presented the preliminary results in Ajaccio. So briefly, we have the field started in 2015 with 10 selection, then 2016 with additional 20 selection, and later on with other 70 selection. So for the first plot, the older one, as you can see here, on the blue one is the prevalence in percentage of the bacterium after 5 years of natural exposure, while the orange one on the right is the percentage of the infected plants showing symptoms. And while the numbers are the score of the symptoms, you can see that only Lecino remain at a acceptable level with a very low score of symptoms. And this is one example. This is our worst Lecino that we have in our field. While some example of score 2, in this case in Frantoio, score 3 also in Frantoio, score 3 in Koroneki, 3.5 in Arbequina, score 4 in, so a few examples. Also, some cases of a score 5, so we did the canopy completely gone. So the second lot of was on 20 selection, and again, almost except Lecino was the only one with a bacterial population that infected only 20% of the plants, but also with a low bacterial population, with Chiku ranging around 28, while all the remaining plants got, when infected, reached the bacterial population higher, so, and the Chiku in real time was an average of 24. This is for all the others.
Donato, you have 1 minute.
Yes, thanks. A couple of them still remain with promising results, but no one is like Lecino. Just reading the conclusion. Even if in some genotypes lower incidence of infections and longer asymptomatic periods were recorded, so far none displayed clear phenotypic traits of resistance. I mean, low prevalence, symptoms, and bacterial population reduced, similar to that observed in the resistant control Lecino. On the other hand, in all experiments, Lecino confirmed our previous observation with lower prevalence and so on. The data obtained so far strongly reduced our initial optimism, showing that the occurrence of genetic traits conferring resistance to Xylella pauca may be very limited in the olive germplasm. The availability of solid and significant availability of a number of resistant cultivar will probably require a long time, and this situation confirmed the need to put in place and maintain all measures to restrain and limit its further spread in olive growing area. Thank you, and I apologize for taking 1 minute more.
Thank you very much, Donato. It is indeed a lot of work to be showed only 15 minutes, but we have some questions for the end of the section. And so I would like to invite Dr. Giuseppe Lima from the Department of Agricultural, Environmental, and Food Science, University of Molise, to present the talk, Evaluation of Alternative Compounds against Xylella fastidiosa subspecies pauca in olive groves of the Salento Peninsula of Apulia. Please, Dr. Giuseppe. Dr. Giuseppe Lima is there? No?
No, he's not there. Maybe we can go ahead and then come back to him. Okay?
Okay. So maybe you can start some questions to Donato.
I'm here.
You are. Okay.
Phone and my phone and video was not allowed. Okay.
Okay. So.
Are you hearing me? I'm here.
Okay, can you share your presentation?
The share function is not allowed. The share function is not run.
Okay, so maybe the technical assistance.
No. Okay.
Perfect.
Sorry. Okay.
Go ahead. Thank you.
My presentation. I go to full screen. Is it? See it? You?
Yes.
Okay, well, good afternoon to everybody. Thank you for to organizer for this opportunity. I will try to summarize years of our research to set up sustainable tools against Xylella fastidiosa pauca on olive in the Salento area of Apulia, southern Italy. In the infected zone of the Salento Peninsula, in addition to search for olive-resistant or tolerant germplasm, there is a strong need to find new effective control tools to preserve susceptible olive cultures and also to improve condition of resistant or tolerant cultures. In the last years, we carried out several laboratory in vitro, greenhouse, and also field experiments with the aim to test several potential compounds, sustainable compounds with the potential activity against Xylella fastidiosa. At the beginning of our activity, we tested different compounds, in particular mineral compounds as macro and microelements, organic compounds, seaweeds, polyphenols, tannins, flavonoids, chitosans, and so other, and some synthetic compounds, as in particular zinc and copper systemic formulates. Several tests were carried out. At the beginning of our activity, we used non-quarantine bacteria due to some difficulty, as you know, to use Xylella fastidiosa because it can be managed only in authorized laboratory. And we, by using different other phytopathogenic bacteria, observed a strong inhibitory activity of some products against different bacteria, and also against, in the latter tests, also against Xylella fastidiosa pauca, the Donno strain, and also against other subspecies of Xylella species, or also subspecies of Xylella, with strong inhibitory activity at concentrations normally used for field application by these products. We also determined the minimum inhibitor concentration of some products, and also in a laboratory scale, we also studied the biostimulant, the potential biostimulant activity of some of these, of the most promising products. Following, we also started with the greenhouse experiments on pot ground olive plants of the susceptible cultures, Xylella, artificially inoculated, preventive treated by our most promising products. And during these experiments, we also studied the bacterial population inside the plants. On this part, further details, you can find further details in a specific presentation that was taken by my co-worker, Carmine Del Grosso, days ago in the session Control of the Bacterium in the Host Plant. Well, we moved to the field experiments where we tested our most promising compounds in different fields of different areas of the Salento Peninsula. Just we have a few time, and I just show you some results of these experiments. In particular, in a field, we applied treatments by some selected natural compounds for 2 years, from 2017 to 2018. Later, we observed at the third year significantly reduction of infection by a systemic copper product, and also results comparable with some of our natural compounds, in particular a compounds SWP based on seaweeds and polyphenols, and other natural polyphenols. When the experiments were concluded, the olive farmer stopped the treatment, and then the following years, Xylella symptoms increased progressively in all the plots. Another, as you can see in this slide, and then without no treatment, after the conclusion of the experiments, we have not more difference, significantly difference from the treatments. Similarly, in another olive groves, we observed good results after 2 years of treatment by natural products, 3 treatments per year. And as in these experiments, no insecticide against the vector were used. And as we clearly can see here, the infections moved from the untreated plots from the treated one when the treatments were stopped. And as in the other field, also here, after the conclusion of the experimental trials, the symptoms were increased quickly in the following years. Similarly, in another field, we observed the same trend with good and significant difference in the year of the treatments, or just 1 year later. But after 2-3 years, also the treated plants decline. Among the products we used, in particular, one of these seaweeds polyphenols, a mixture of seaweeds and polyphenols, was also applied in a field on the soil, other than by spraying on the canopy. And this is an overview of the treatments. Just the distribution. The distribution was under the canopy projection on the soil. This is an image of the last olive groves after the treatments were stopped 2 years later. And as you can see, we have some differences on the treated plants, but anyway, the symptoms of Xylella are spread. This is an experiment we carried out, a very interesting experiment carried out in a nursery on Lecino plants, where we observed that by using treatments per year, in particular in 6 months from March to September, on plants treated by seaweed polyphenols, we observed an increase, a consistent increase of the plants height and number of shoots, as you can see in comparison with the control. And the treatments applied on the soil, on the pot surface, were more effective than sprays on the canopy. Based on the previous our field experience, we now are tested an integrated strategy by combining together different products, our best products, on the canopy by spray the canopy, and also by soil treatments, and applying also insecticide when in the phenological moment where the vector is spreading from weeds to on the plants. In these trials in different areas of the Salento, we are also monitoring the effect of the treatments on the Xylella population inside the plants. In conclusion, our experience said that in the absence of control measures, agronomic, biological, or chemical, in the infected area of Salento, the olive trees are destined to progressively decline and die if no treatments are applied. The new formulations were investigated are not miraculous. Anyway, reduce the progression of Xylella fastidiosa symptoms appreciably. However, they are not able to eradicate the pathogen inside the plants if singularly applied. Now, in different olive groves, we are studying an integrated approach based on the combination of soil and canopy treatments with the most promising products, together with the preventive agronomic and chemical measures against the vector. In the absence of sustainable alternatives, our approach could be useful to avoid the complete loss of traditional olive cultures, deserving also particular attention to the high number of monumental trees, centenary and even millenary trees, characterizing culture and traditions of the territory and its inhabitants. Our integrated approach could be a valid tool to manage also Xylella fastidiosa resistant, tolerant olive cultures, for example, Lecino and Tolosa. We have, don't forget that these are resistant but not immune cultures to Xylella. And to obtain appreciable results, the control measures should be kept over the time, and not just for 1 year, 2 years, or just 1-2 treatments per year, and applied in a wide areas and not only at single farmer level. The management costs are often not sustainable for olive growers, and then regional, national, and European community governments should better support olive growers. I would like to thank my co-workers of the plant pathology team of the University of Molise, and Donato Bosch, Maria Saponari, and Pasquale Saldarelli, and co-workers from the Bari CNR, and also some projects that supports partially our research, and Stefania Loretti from CREA in Rome for providing us with the non-quarantine pathogenic bacterial strain. Thank you for your attention.
Thank you very much, Dr. Giuseppe. And so we are very late, but I think that we have some questions for Donato. And I would like to Donato answer some of them. And the others we are going to send directly to the research, and then they can answer to you later. But so, Donato, do you have any genetic data on the olive varieties tested that could explain the susceptibility or resistance? In addition, do you have an idea of the genetic diversity of the olive varieties tested? If so, is this correlated with your results?
Well, concerning the first part of the questions, there are my colleagues Pasquale Saldarelli and Annalisa Gianpetruzzi mostly that have done already. They are working in this field, and we already published a few years ago a paper with the transcriptome analysis that has been done comparatively between Lecino and the susceptible cultivar Oliarola. According to this, I will say preliminary data, seems that there is also some genetic mechanisms that is, in any case, is not monogenic but it's polygenic, because seems that there are genes that are correlate and seems to have a role in the degradation of the cell walls. So in different cultivar, resistant or not resistant, they are differentially expressed. So this is one line, but again, there is still a lot that is not known on this part. There is also, on the other hand, probably also some explanation that is related also to the anatomic structure of the different cultivar, because there is a, if I'm not wrong, a paper that has been published by colleagues from the University of Lecce, Luvisi and co-workers, associating the different sides, if I'm not, of the xylem channel, or the xylem tubes that of different sides between Lecino and Oliarola. So this may have also a role. Concerning the second question, I think to be not the best one to have the proper knowledge to answer to this. This should be, I'm not a genetist of olive, but I believe that we still have too little to answer to this, because as I show you before, the great majority of the olive germplasm seems to not show any characters of resistance.
Okay, thank you, Donato. And are you doing some trials using FS17?
Well, for FS17, we are not doing anything special in the laboratory, at least in our trials, because we are following that there are several fields of FS17 in the highly infected area. So we are following we are monitoring these plants in that area, and as Maria shown on Monday in her presentation, FS17 also as well as Lecino, they confirm to have characters of resistance and to behave satisfactory when properly managed. But when infected, they have a higher requirement of management, because if you manage Lecino FS infected with minimal management, I mean without water for irrigation, without regular pruning, they also start to show problems.
Okay. Okay, thank you, Donato. And so one question for Dr. Giuseppe. And so have you done some study about the economic cost of using this product in the field?
Well, we are evaluating this aspect with the economists, but it is an important concern. It is obvious that in the absence of a production, in these last 5 years, 6 years, in the Salento years, in the Salento area, the production was drastically reduced. In this condition, olive grounds are in loss, in economic loss, and then we are not prone to sustain cost for the control.
Good afternoon. Welcome back after the short break. We continue with this session on sustainable management strategies. In this second block, there are 4 talks. One will be a 15 minutes talk, and the last 3 ones will be 5-minute talks in the frame of the Young Researchers Initiative. So now the next speaker is Stefan Kompant from the Austrian Institute of Technology. And the talk is entitled BioExo, a new Horizon 2020 project on biocontrol of Xylella and its vector in olive trees for integrated pest management. So Stefan, the floor is yours.
Thank you, Antonio. Good afternoon, everyone. You have seen this logo of BioExo since Monday, and I would like to explain you what is this project. This project, in fact, started previous year in May 2020. And this is a grant from European Union, just the BBIGU, the Bio-Based Industry Joint Undertaking. And it's a grant of 6 million euro for 5 years. We are 11 partners with industry, SMEs, academia, but also farmer and farmer association. The project is coordinated by RTDS Group. It's an association in Austria. We have as partner Globachem for pesticide and biopesticide from Belgium. CNR, of course, we have seen a lot of colleagues from X Factor. Some of them, they are also in BioExo. We have a farm in Mallorca, Emirate, CRSFA, Vivenza Don Giovanni from Italy, University of Antwerpen, Domcat, this is an SME from Spain. And as other partner, we have also Azaja from Spain, us from AIT, and the University of Sevilla for toxicology test and Access Bio as an SME from Slovenia. What is BioExo? In fact, we want to demonstrate the use and to increase the production of biopesticide targeting directly Xylella as preventive approach or curative approach. But also
Hello, Stefan. Sorry to interrupt you. You should have a window upon your presentation. It should be a window of WebEx. If you can move it aside, I don't know, at the bottom or.
I know I don't see it. Sorry. A window.
Yeah, you're on it with your mouse. If you can move this window just aside or at the bottom of the screen. Okay.
I know what you mean. Yes.
Yeah. Because we see a green patch. Okay.
Okay, Jessica.
Okay, thank you. Thank you.
And so we are developing biopesticides from the component in research and development, but also making formulation, upscaling of the production of this biopesticide, making field trial, and we want to be at the end of the 5 years of this project close to the market. Our long-term objective or to first to secure our orchards, to reduce the Xylella pressure, to reduce the disease severity, and also to protect in long term orchards, but also to protect jobs that have been lost because of Xylella. More than 100,000 jobs have been lost. And we want to protect our societal things, but also our cultural heritage in Europe. And to ensure, of course, the continuation of olives and almond cultivation. For this, we are testing different solutions, bacterial strain as biopesticide, microbial metabolites, plant extract, entomopathogenic fungus. We have not been starting from scratch. In fact, we are developing already some product at technology research level 4 to 5 with some in vitro test on the greenhouse condition, and there was also some few tests in the field. Within this project, we have different work package. First work package is led by José Manuel Garcia Madero from DOMCA, is to develop biopesticide targeting Xylella based on bacteria or plant-based extract. We are making tests in Apulia, but also in Mallorca for olives and on almonds in mainland Spain. We try to identify the best application method and the best products in a small field trials, as curative or preventive, via foliar spray, endotherapy, or irrigation. And we are monitoring this each 6 months by qPCR and also the symptom. For the work package 2, this is led by Lisbeth Swartz from Globachem, from Belgium. We aim to develop biopesticide targeting the vector, to identify the best application method, the best substances, and also to confirm efficacy, to optimize the dose range, to apply it at the good rate, timing, and interval. In work package 3, this is very important work package also, is to make formulation and upscaling of the product. You know, for field application, we need not 5 liters, not 10 liters, but 100 liters, 500,000 liters. And we make upscaling of the production in industry from lab scale to industrial scale for production for field and also to make control of the bioprocess. Of course, we are not using something that is very costly because we want to reuse bioresource to have economic viability of our product. This is a very important work package. But of course, we have also work package, I think you know Pasquale very well, for large-scale validation of our product. For Xylella control, to reduce the bacterial spread, the load of the pathogens in Apulia, mainland in Spain, but also in Mallorca, to combine the strategy of applying biopesticide in an integrated pest management approach with weed removal or combination of biopesticide targeting Xylella or the insect Philainos plumarius. As another work package, we are understanding how this biopesticide can work. So this is the mode of action. If the biopesticide can directly target the Xylella as antagonism, or boosting plant defense reaction, or colonizing plants inside the xylem vessel at the same place as the Xylella, or maybe at the distantial part of the plants by secretion of some substances from our biopesticide. We aim also to study the effect of our biopesticide on the colonization process on the insect, meaning that not only with fungi killing the insect, but also if our biopesticide that normally targets the Xylella, if they can also colonize the insect and be present also at the same time in the foregut of the insect, where the Xylella can be present. This is mode of action. This is support for product development and registration. And we have also an important work package on toxicology. We are making life cycle assessment of our biopesticide. We make toxicity testing, evaluation to have the best product and that we can use for the environment. That there is no toxicity for human, animal, environment, but also to protect our bees, our pollinator in the organ, and to have the selection of the best solution for this. You may have seen also us on Twitter, YouTube, Facebook, Instagram. We are doing also dissemination and also exploitation and communication of our results. This is led by Adela Naku from RTDS Group. We disseminate here on social media, but also we are establishing project community through the dissemination, exploitation, and communication activities, and to manage also intellectual property of our biopesticide, because we are working with industry, we are scientists, but also there is partner from industry, and we are making IP of our product before to release them close to the market or to the market. Lastly, you know, when you are doing a European project, it's very important the management. This is coordinated, like the project, by RTDS Association. Me, I'm just the scientific coordinator, putting the science inside. And for the management, we need to coordinate, of course, organize a meeting, also administrative tasks to implement the common strategy, financial management, data management, and also to communicate with the funding authority, BBIGU. This is done by Stefan Webb, and is doing today, for example, uploading 5 deliverables of BioVexos that we should send today. So this is a lot of work. Today, I could not speak too much about the project, just a project overview. As I told you, we are working with industry and farmer. We are testing different kind of biopesticides, but I wanted to show you the overview, the objective of this project. And if you have any question today, of course, you can ask me, but we would like to hear from you. Just write us at biovexo@rtdsgroup.com. And if you want to follow our update, we put some nice video on Twitter, LinkedIn, YouTube, Instagram. And I would like to thank you for your attention. And you can see also we have a nice website, www.biovexo.eu. Most of this work present today come from this website. I would like to thank you very much for the opportunity to speak. And if you have any question, of course, if I can answer, you're welcome.
So thank you very much, Stefan, for this very nice overview of the project and all the people and working package involved. Before moving to the Young Researchers Initiative, I would like to remember the audience that you can post your questions on the Q&A section of the WebEx, and we will collect them. And after the talks, there will be a question and answer for all the speakers in this second block. Okay, so now moving to the next talk by Manuel Anguita Maeso from the Institute for Sustainable Agriculture, CSIC, in Spain. And the title of the talk is Culture and Metagenomic Approaches for the Identification of Olive Xylem Microbial Communities as a Biological Control Tool to Cope Against Xylella fastidiosa infection. So Manuel, the floor is yours.
Thank you, Antonio. I hope everything is fine. Hi, everyone. I would like to thank for selecting me as a Young Researcher Initiative, and I would like to share with you this work. No? As you know, the xylem-inhabiting plant pathogenic bacterium, Xylella fastidiosa, represent one of the major phytopathological threats to olive growth worldwide because of its devastating effect on agricultural yields and high tree mortality that causes a profound socioeconomic and environmental impact. Focusing on Brazil, the olive leaf score associated with Xylella was first reported in 2016 by Dr. Elbecio, specifically in the Minas Gerais state, and since then has been spreading to other regions. In order to fight and hopefully defeat this pathogen, endophytes are a promising strategy because they play an essential role on plant growth and its physiological status, but they can also act as an innate natural defense to cope against infection by xylem-inhabiting pathogen. The experience and knowledge I'm gaining in my PhD with the study of another xylem-inhabiting pathogen, Verticillium dalium, represent a good starting point to apply different strategies against Xylella fastidiosa. So in previous research, we have shown that olive xylem sap harbor a unique source of bacterial species, many of these which can be cultured in artificial medium mimicking the xylem sap composition, and then transplanted into olive planting stock, modifying its xylem microbiome. To this point, I would like to encourage you to see my e-poster, Broad Media Cultivation of Olive Xylem Microbiome for Cultivated Olive Trees, to find more information regarding this issue. You can go directly to this code. So the objective of this work was to characterize the olive xylem microbiome associated with Xylella fastidiosa infection through culture-dependent and independent techniques to find a specific bacterial species associated to healthy tree status as a tool for identifying potential biological control agent for this pathogen. We focus we selected 2 field locations in Sao Paulo state in Brazil that differ in 500 meters of altitude. We focus on 4 olive genotypes, Coroneki, Arbosana, Arbequina, and Grappolo, and we compare symptomatic infected trees checked by qPCR with asymptomatic non-infected trees. Also, we collected sample of stem and root. With the bark, this xylem tissue, we sterilized and we made we obtained an extract that we use for methodological approaches. On one hand, we follow culture-independent analysis. We choose R2A medium because we observed in our previous study that this medium recovered a major amount of endophyte. And in this case, we compare this medium with plant extract that came from filtering and mixed root stem. And on the other hand, we followed a culture-independent analysis. So we extracted the DNA, and the next steps are still in progress, that are the library of 16S and ITS, and the identification of the amplicon sequencing buyer. Some preliminary results that we obtained, we obtained a total endophyte, 308 endophyte, and we observed that the main differences based on unit formation colonies were 2 ecological niches, higher in root than in stem, and followed by the field location, higher in lower altitude, and the Xylella infection, higher in non-infected trees. According to genotype, Grappolo displayed the higher UFC, followed by Arbequina and Coroneki that were quite similar, and finally Arbosana. If we structure this variable study according to the genotype, we observe that Arbosana and Grappolo follow the same tendency in F2 field, but in the stem of no inoculated plant in Arbosana was higher, and Grappolo follow a distinct behavior. Also, if we observe in the root of, according to Xylella infection, we observe huge differences in Grappolo, in Coroneki, in Arbequina, but this difference was not so visible in Arbosana. So, conclusion, the result indicated that the difference in the frequency of microbial community depending on the type of plant material analyzed, the environmental location, and the Xylella fastidiosa infection. The next step will be focused on identifying the isolated bacteria using SenGen technology and the total microbial communities using NGS approach. And overall, this result will help to expand our knowledge of xylem microbiome community, and more importantly, to identify xylem inhabiting microorganisms with potential to combat this harmful bacteria. Thank you for all the institution and project that give me the chance to present this presentation, in particular Carolina Foundation for the research stage. Thank you for my group in Spain, and a special thanks for Helvecio and for our amazing trip around Brazil. Thank you.
So thank you very much, Manuel, for this interesting talk and showing very nice images from Brazil. I'm sure Alexander liked being at home in the conference now. Okay, so let's move to the next presentation, also in the Young Researcher Initiative. In this case, the speaker is Marwa Mourou from the Dipartimento di Scienze Agrarie e Forestali, Università degli Studi della Tuscia in Italy, and the title of the talk is Screening of a Novel Biological Control Agent Targeting the Phytopathogen Xylella fastidiosa. So Marwa, the floor is yours.
Good afternoon, and I'm really glad to be accepted for the program of Young Research Initiative and to have this great opportunity to introduce my contribution. Briefly, in 5 minutes, I would like to present my research work titled Screening of a Novel Biological Control Agent Targeting the Phytopathogenic Xylella fastidiosa. So let's get started with some general information. Unfortunately, the situation of the olive sector in Italy has been changing from 2013 by the outbreak of olive decline syndrome disease. And just a recent estimate suggests that there are 4 million olive trees that were heavily infected or dead, which equal to 10% of loss of Italian olive oil. And in spite of the pertinent research effort devoted to control the disease, so far there are no control measures available to totally eliminate the bacteria from diseased host plants. Therefore, the aim of our study is screening of potential antagonistic bacteria on the phyllosphere of 8 different host plant species. Okay, starting from collection of sample and isolations. So sampling of symptomatic and asymptomatic leaves from 8 different host plants of Xylella in the
So maybe technical support could help Marwa with maybe some connection problems.
An dilution series was made from each solution, and 100 microliter of each solution was spread on agar medium. After day of incubation, we obtained the following graphs. So 89 isolates were obtained from 8 different host plant species. As shown by the graph, differences in the population size were observed between the different hosts. The highest one was recorded for Polygala myrtifera, followed by Olea europea, then for Prunus dolces and Aviomore, more or less the same, and the lowest one was for Laurus nobilis. Moving for the screening of antagonistic activity in vitro, that was carried out by dual culture assay. The bacteria were co-plated as following: 3 drops of Xylella suspension 10 to the 6 colony forming units were placed at the top of each plate, allowed to flow down to the opposite side of the plates by generating 3 rows, as shown by the picture A. Then an aliquot of 5 microliter of suspension of the potential biological control agent was placed on the top of each row of Xylella fastidiosa on the plates. Then, after day of incubation, the antagonistic activity was detected, and the growth inhibition was measured in millimeter. The following graph represents the results of the growth inhibition assay. 12 out of 89 isolates were able to reduce the growth of Xylella fastidiosa with the appearance of a clear zone that goes from 4 to 24 millimeters. The highest one was recorded for the isolates BA69, then as shown by the picture B, then the isolates BA103 and BA102 as shown by the picture C. Subsequently, the 12 antagonistic bacteria were submitted to biochemical tests, fluorescence, Levin, gram stain, and oxidase tests, and were identified by analysis of 16S region. The following table summarized the results and revealed different bacteria species that belonging to Pseudomonas graminis, Dilthia acidovirans, Cryptobacterium flacinfections, Pontio agglomerans, Microbacterium oleifera, oxidans, and Philosporiae and Stenotrophomonas rhizophila. As I mentioned in the previous slide, the highest one was recorded for Pontio agglomerans, antagonistic activity, followed by Stenotrophomonas rhizophila and the Microbacterium species. As a conclusion, 12 epiphytic bacteria were able to reduce the growth of Xylella. Several tests are ongoing to understand their characteristics and their mode of action, such as antibiotic, lytic enzyme, siderophores, and antimicrobial activity. And we hope to go also to identify the specific inhibitory compound that could be secreted by these antagonistic bacteria. And as a future work, the antagonistic efficacy of the most efficient isolates in vitro needed to be tested also in vivo. Just to mention at the end that this work was made in collaboration between the University of Tuscia and the Mediterranean Agronomic Institute of Bari. And thanks all for your attention.
So thank you very much, Marwa, for this very interesting and comprehensive work. So we are well on time, and we will move now to the next speaker, also in the Young Researchers Initiative. Is Luis Alejandro Moy from Institute of Food and Agricultural Technology at the University of Girona in Spain. And the title of the talk is Peptides of Synthetic and Microbial Origin with Antimicrobial and Antibiofilm Activity against Xylella fastidiosa. So Luis, the floor is yours.
Good afternoon. First of all, I would like to thank you for the opportunity for doing this presentation. And today I would like you to present some of the results we have obtained so far in a project that we are currently doing at the University of Girona in the CITSAP group in collaboration with the LIPSU group. So as everybody knows, Xylella fastidiosa is a plant pathogen which forms biofilm in the xylem vessels of infected plants. Usually, this biofilm is associated with an increase in resilience against antimicrobial compounds. So at the moment, we don't have any efficient treatment to attack or to fight against Xylella fastidiosa infected plants. So there is a need to develop new treatments, and one potential candidate compounds that we are actually focusing in this project that we think that are potential candidates would be antimicrobial peptides. So in this work, what we did was to obtain peptides from two different sources by synthesizing them and from microbial origin. In the case of synthetic peptides, we did first a search in the literature for peptides that were previously described to have a good biological profile against gram-negative bacteria. And we selected some of them. And then we proceeded to synthesize them, purify, analyze, and characterize them by HPLC and mass spectrometry. In the case of microbial origin peptides, what we did was to do a screening to our microorganism collection by PCR, and we searched for genes related to enzymes that are related with the synthesis of antimicrobial peptides. Those strains that were interesting, we proceeded to culture them and obtain their supernatants, and we tested their activity. If they were active, we proceeded to fraction these supernatants to try to isolate the active compound responsible for the activity. So for all the peptides and supernatants that we have obtained, we measured their activity, specifically the antibacterial activity, using a contact test coupled with viability qPCR and plate counting. And we also measured antibiofilm activity using crystal violet methodology. We also assessed the toxicity of these peptides and supernatants using determining the phytotoxicity on tobacco leaves and also the hemolytic activity on horse blood erythrocytes. So here I show you some of the results we have obtained for the synthetic peptides. Here you have the peptides that we have tested. We have synthesized around 31 peptides so far, and are organized for structural families and are here located in boxes. The colors, what they mean is that the stronger the color, the better the biological profile. So in case of activity, the stronger the color means the higher the activity, and in the case of the toxicity, the higher it is, the color means that it's less toxic. So in general, what we can see from all the peptides that we have tested is that we have identified some peptides that have really high antibacterial activity. Most of the tested peptides showed a high antibiofilm activity and many a low moderate toxicity profile. I would like to highlight three peptides, which are the best ones, which would be B2, Maganin2, and I4, as they have a really high antibacterial activity and moderate high antibiofilm activity. And as I mentioned, they are not, they are moderately toxic. So in the case of peptides of microbial origin, as I mentioned previously, we screened our microorganism collection that we have, and we focused on three genuses, which are Bacillus, Pantoea, and Pseudomonas, and we searched for genes of enzymes related with the synthesis of ribosomal peptides and non-ribosomal peptides. The strains that were the most interesting, that were it was possible that they were synthesizing these peptides, we proceeded to do to culture them and to obtain their cell-free supernatant, which we tested again using a contact test by plate counting and viability qPCR. So here you can see the positive control without treatment, and here with the cell-free supernatant. So the ones that were active, so no growth was observed, we proceeded to fraction these supernatants to see we were able to isolate the compound responsible for this activity. And we did the test to see which fractions were active. So just to sum up a little bit, in previous works, we have identified that some peptides had antibacterial activity against Xylella fastidiosa. An example would be BP178, which has been tested and you can find the results in these papers. And in the current project that we are working right now, our main objective is to search for new peptides directed toward key targets and processes to control Xylella fastidiosa. And specifically in this work, we focused on antibacterial activity and antibiofilm activity, which we have tested in vitro. At the moment, we are starting to do some plant assays with compounds, or the active compounds that we have found. So just to conclude, from the 31 peptides that we have synthesized so far, we have found 2 which a really good biological profile, which means that they have promising high antibacterial and antibiofilm activity in vitro and low moderate toxicity. And we also have identified some cell-free supernatants which were active. And at the moment, we are also working on trying to isolate the active compounds and trying to identify them. So to end, this project was funded or is being funded by the Spanish government and the European Union. And also I would like to thank all my colleagues from the CITSAF group and the LIPSO group. And thank you very much for your attention, and I hope you have found my presentation interesting.
So thank you very much, Luis. Very interesting presentation and very new approaches for the control of this disease. So now we are perfectly on time, and we'll start with the question and answer part of this second block. So I will ask all the speakers of this second block to turn on their camera, so I can just start to send questions to all of you. Okay, so following the order of the speakers, the first question is for Stefan on the BioVexo project. From a, let's say, scientist or developer perspective, you are working to put new tools in, let's say, in the market for solving these problems, particularly in this case of Xylella. I would like to have your views on the current system for registering of these, let's say, products or biocontrol agents, especially when we are dealing with a plant health emergency, like is the case of Xylella. So if you can maybe follow up on that point, will be very interesting, your perspective.
Yeah. Thank you, Antonio. Yeah, this is a very good question. In fact, there is a lot of things to do before to get the registration, and also to have good laboratory practice, a lot of tests, the toxicity. And it's cost a lot of money also for industry to do all this kind of tests, and we need after to have an approval for that. And at the end of the project, we will be close to the market, but we will need also to make some other tests after. We could not arrive in 5 years, starting from previous years, with already product ready to sell to farmer. But they will be very well constructed, and we will be at TRL 7, 8, close to the market. But it takes time for registration of biopesticide. It takes time, but also it takes a lot of money for this. And of course, we are making also tests on the where the plants are on the containments and the occurs. When there is an eradication zone, we cannot test our products, unfortunately. So it's there is a base for this, but we have different pedoclimatic conditions where we are testing in Apulia, but also in Mallorca and mainland in Spain. In mainland in Spain, for 1 month, for example, we got a program because it was an eradication zone. So we are just testing as a preventive approach in this area. Yeah.
Okay, so thank you very much for your comments on this. And changing a little bit the order, now the question is to Marwa. The question is from the audience is, colonization dynamics may differ significantly from philosphere to xylem network. As you focus on epiphytic species, are you confident with their possible efficacy against Xylella fastidiosa in planta?
Establishment of the biological control agent on the aerial surface of a plant, it's really a challenge, especially because I'm working on xylem bacterium that colonize the xylem. But this is a point I will take it on consideration, because as I see, my results like Panchia agglomerans and Stenotrophomonas rhizophila are bacteria that are already known to be also endophytes and can live on the sphere. But it's a fundamental step before I start the in vivo test to test their ability to colonize the xylem vessel and try to detect them after colonization far away from the point of colonization, to be sure that really good. So this is an interesting point that for sure I will take it in consideration.
So thank you very much, Marwa, for your answer. And now a step, we go to Manuel, because it's a kind of related topic on this. So you are working with all of these microbiological communities inside the xylem. Xylem usually is under negative pressure, especially in these quite large plants like olives. So do you think this is making selection pressure on the microorganisms that are there? And as we were discussing in this previous question, how this can affect also the possibilities of using these organisms as biological control?
We extracted the xylem sap with a Scolander chamber device, and we increased the pressure up to 40 bar. No, so we extracted all the sap of the xylem sap. In some cases, we extract all the bacteria, but some bacteria maybe can be sticky in the xylem vessel. And maybe we are losing kind of this information. But on the other hand, as we apply so much pressure, so we remove all this sticky. We try to move all this bacteria from the xylem vessel and recover everything as much as we can.
Okay, thank you. Now a question for Luis, also a question by the audience. By PCR, are you screening for already known peptides in new bacterial species?
Yeah, well, we are yes, we are screening for already described peptides in the genus. For example, Bacillus has been described to produce a series of compounds, so antimicrobial compounds. So we searched for these enzymes related to the synthesis of these products on the strains that we have isolated or that we have in our microorganism collection. They are not new species per se. It's like we have isolated them and we have identified them, and we have them in the university here.
Okay, thank you. Now also a question by the audience to Stefan. This is quite very specific question. I'm sure there are some maybe logistic things related, but I will. So why you did not select Ibiza olive Pauca for your trials in Spain? This strain is more similar to Pauca from Italy and the most aggressive for olives in Spain.
Yeah, see, it's a good question, but there was a connection between a farm in Mallorca, we know, with an industry from Spain, and it's why we have gone to Mallorca for this. Yeah, this is just logistic.
I understand.
It's a very good question. You know. So, and we wanted also to test the different climate, soil, and we did not think about Ibiza, sorry, and we did not have a connection. But maybe for the future, for the next few years, to make some tests also here. Thank you.
Okay, thank you, Stefan. So now following the order, a question to Manuel again. During this conference, we have seen the major epidemiological and also biological differences of Xylella and the diseases that it causes from Brazil, for instance, and in the case of the Mediterranean basin. I guess these differences could be also in the case of the microbiome you are studying. Do you think that your results from Brazil can be directly extrapolated to the situation in the Mediterranean, or maybe you already have some information from the microbiome in Xylella infected plants in the Mediterranean.
Well, in Brazil, talking about Brazil, we are now running all the sequencing analysis to get the information in microbiome there in Brazil with the Xylella infection. My previous result here in Spain, we observed some differences in the environmental location in Andalusia, in the south of Spain. But what is most important in this microbiome differentiation is the first the plant niche. According to the plant, we observe significant differences in the rhizosphere, in leaves, in seed and sap, in different parts. No? But then the environmental location have a strong effect in this differentiation, the climate. No?
Okay, thank you very much, Manuel, for your question. Now there is a question also by the audience to Luis. Once a good antimicrobial peptide is identified, what will be the strategy to make possible to produce and use it at an affordable price?
Yeah,
recurrent question, I think.
Yes, it is a recurring question. In the case of synthetic peptides, as we all know, it's quite expensive to produce, like chemically. But we actually, in the THIT subgroup, it was published some time ago. I actually have it right here in 2017, a paper in which we were able to produce one peptide that I mentioned, which is BP178, in rice seeds, and so rice seeds were used as biofactories. This would cheapen the production costs of creating this peptide. And for the case of microbial origin peptides, it's more easier to obtain because microorganisms are easy to grow, and since these peptides are produced outside of the cell, we can obtain the supernatant and easily isolate the interesting compounds that we want to use to treat the trees Xylella fastidiosa.
Okay, thank you, Luis. So if may I ask also a question following on that issue. I know that your group is doing, let's say, implanta test with these compounds. My question is also the extrapolation of these results to, let's say, adult plants, like in the case of olive trees or almond trees with, several tons of biomass in the tree. And not talking about the cost, I'm talking about the efficacy. So if you can maybe provide some guess on that, let's say, a scale from small plants in the greenhouse to large plants that are the ones in the outbreak areas.
Well, how we explain this? Actually, we are working on greenhouse conditions, and we usually use plants that are not that old. So I'm not sure how I would extrapolate to an adult plant, because this is like the next step that we would be looking for. First, we are doing like the first tests in like young plants, because it's more manageable to treat and to apply the treatments and see the effects. And once we have obtained these results and these were like interesting enough, we would pass to the next phase, which would be trying to do field trials, maybe, for example, in Mallorca or in Alicante, which we have the outbreaks, and try to see if these methods are efficient enough in adult plants, and we are able to control Xylella fastidiosa.
Okay, thank you very much, Luis. And now come back to Stefan about the different strategies that you are testing in your project. You said they are preventive and curative, let's say more therapeutic strategies. Which of them do you think is more likely to be effective for the preventive approach or more the curative therapeutic approach? Not easy question.
Yeah, it's a good question, but not so easy, because we have done also preventive approach since 1 year, but for the moment we are waiting Xylella symptom. Curative, we got some good results, and on one occurred, and another one it was not positive. So we need to test a lot, and to test also, I forgot to say, on different variety, more susceptible, less susceptible, and in different place. The best will be that it works as a preventive as and curative. We'll see. We are just at the beginning of the project, just after 1 year, and we are testing a lot of different products, different place, a lot of plants. Huh? So, yeah.
And also, sorry for following up the question, but as the comment of Luis before, do you think the target will be better the vectors? I'm talking about the efficacy, not talking about cost or feasibility, but the target on the vector of these new compounds, or the target in the plant as kind of against the bacteria itself?
This is also a very good question. I think both, because if we can target the Xylella vessel with some microorganism at the same niches where the Xylella is present following inoculation by the subfeeder, it could be also a strategy. But of course, if we reduce the vector before it's going on olive plants, it will be also a very good strategy. But we need, in my opinion, to do several approaches like this. And especially also for IPM strategy to target the vector, to target the Xylella of plants, because the plants are already infected. Even if you target the vector before, there is already symptom because it has been transmitted before you put some product against the vector. And also to make some weed removal, and also to combine some approach with a more resistant cultivar, or I would say less sensitive cultivar. So it's a different approaches like this. With one approach, I don't think that we will solve a problem, but by combining different approaches, we can maybe reduce, and I think so, reduce the load of pressure in our orchards. It will take time for this. It's a lot of tests to do, and to do different approach with different partner, because there is some people specialized in one part of the topic and some other part.
Okay, so thank you very much. I don't see we have additional questions from the audience. I don't know, maybe Alexandra, we can close the session just a few minutes before the schedule, so people will be with a slightly longer coffee break. Maybe we want to say a few words to close this session. From my side, I would say that a very interesting session, very new approaches. I think people is working really hard on that. We know that control measures that are now in place by the reduction of elimination of infected plants, control of the vectors are of high impact, social economic impact, but from the session I see that there is still more time needed to become realities, many of these things, but I think there is very a lot of hope on these things, so really encouraging to see all of these results that hopefully in the near future will be implemented in the field. So I don't know if Alexandra, you want to say something to close?
Yeah, I'd like to comment some points. So I was reading here, and I agree with Giuseppe when he said that there is no miracle products to control everything. I mean, to control the bacteria, control the vector, and control the population of the bacteria in the plants. And so I really agree that we can use different approaches and put all your eggs on the baskets, and so to try which one we can use to management the disease in the fields. And so in Brazil, we have this disease for a long time, that's CVC. We are dealing with the bacteria. This disease is not a big problem in Brazil anymore. Now in citrus, we have the HLB or greening that's more aggressive. And so I don't know if you remember, but I still work on a product, on a molecule that is N-acetylcysteine to control the Xylella. And so we are having, yes, good results in the field. Now we have one product that we are trying to expand in the fields, especially now for greening, because the problem we have is, but it's not a control We are not going to finish the bacteria completely in the field. We have a tool, one more tool, to deal with this bacteria and now decrease the economic damage that the growers is having with this disease. We can produce fruits even though we have the bacteria there. And so the idea is to decrease the population of the bacteria, decrease the vector, and put some microorganism that is microbiome to improve the plant, to improve the plant response to the bacteria. So there are a lot of things that we put together. I think that we have a good tools to deal with the bacteria. And so I really believe that we will not have only one thing that are going to control everything, but combine science, combine good results together, and then apply in an integrated manner to control the disease, to control the damage that the disease is causing to the growers and to the olive or citrus or grape production. So that's why, and so I'm very enthusiastic with all the results that was shown here. So it shows that even though we have a company or students or researchers, everybody is working to try to control this disease. And every good researcher is very welcome to try to deal and to understand and to deal with this disease to improve the agriculture in our world. So that's it. Thank you very much for the brilliant presentation that I could see today.
So thank you, Alessandra. Thank you to all the speakers of the session today. And just remember to the audience that we will be back here at 4 o'clock with the session on priorities for research, science support to stakeholders. So it's probably kind of corollary of this conference. I expect it's going to be very, very interesting session. So thank you very much again to all of you and also for the audience for being here this afternoon. Thank you.